everyone. Usually RDT is thought to be about adding useful genes to the host genome so as to acquire certain special properties. But today we shall discuss another unique and useful approach which is quite opposite to it and is called gene subtraction. This involves the use of tools and techniques of genetic engineering to subtract or suppress the expression of some unwanted genes. Here, rather than removing a gene, which is tough, time-taking and expensive, some other strategy is intelligently applied which is much simpler to execute. The underlying principle is to clone a synthetically prepared oligonucleotide in the host genome. This synthetic DNA fragment codes for an RNA that is complementary to the sense RNA, thereby rightly termed antisense RNA. The prerequisite to do so is that the target gene sequence should be known so that the antisense nucleotide may be successfully synthesized. Therefore, as the target gene is transcribed, this cloned gene also undergoes transcription. And being complementary, both transcripts mutually hybridize, thereby not letting the ribosome bind to the sense RNA for the initiation of translation and hence product formation. This technology was developed by Dr. Hal Weintraub et al. In early 1980s. Although simple, this approach is very logical and powerful. It works well and has been used to advantage. Recombinant plants or animals developed in this manner also by definition fall in the category of genetically modified organisms. Let's understand this approach using an example. The enzyme polygalacturonase or PG is responsible for fruit ripening as it converts the tough pectin present in unripe fruits into simpler forms, thus softening the fruit tissue. It was noticed that tomatoes would start spoiling due to ripening and softening even before they reach the customers or consumers, leading to immense losses. To handle this, in 1994, Calgene, a California company, brought the first genetically engineered crop to the market, the FlavrSavr tomato. TI plasmid vector P-bin19 carrying cauliflower mosaic virus promoter was chosen for this cloning. The recombinant vector was used to transform Agrobacterium tumefaciens, which in turn was allowed to infect tomato stems. By executing this, the company's scientists were able to reduce the enzyme production that was responsible for turning the tomatoes over soft, squashy, and spoiled very fast. FlavrSavr tomatoes, being a GMO, when appeared for sale in supermarkets, had caused an enormous media stir at that time. Thus, Antisense RNA technology by gene cloning was applied to tomato plants for delayed ripening. Some of the antisense RNA hybridized on its own with the sense RNA of the PG enzyme in vivo. This resulted in much lesser amount of the enzyme, slowing down the softening and ripening of tomatoes, hence preventing their mass decay and enhancing their shelf life. FlavrSavr tomatoes were large in size, blemish-free and exhibiting delayed ripening. This was certainly a great demonstration of antisense technology or gene subtraction. It's worth mentioning that even if the antisense RNA provided to somewhat silence the target genome is even smaller in size, still it can partially hybridize to it and block its expression. Another important point is that the sense RNA is never blocked 10% since some of them naturally steer clear of the antisense RNA molecules within the living host cell. And of course, this is desirable as only delay in the ripening was the goal rather than failure of ripening. If the recombinant tomatoes remain raw and hard forever, they would be useless and the whole purpose would be defeated. Although this approach does suffer from certain limitations, still it was a successful demonstration of a viable idea with immense potential. That was the real take-home message. Through the understanding of development of FlavrSavr tomatoes, this technology has found immense use in agriculture, medicine, among many other fields. Since antisense technology focuses on preventing gene expression, it has most widely been applied to cancer gene therapy. Antisense drugs are being researched to treat cancers including lung cancer, colorectal carcinoma, pancreatic carcinoma, malignant melanoma, and also diabetes, Duchenne muscular dystrophy, asthma, among many other dreaded diseases. So guys, we hope that this RDT strategy has become ingrained in your memory. We'd love to hear your inputs and feedback of our work in the comment box. Our mission is to make you understand all such concepts. That's why we describe things in a simplified and laymanish manner. We thank you for your graceful acknowledgements and encouragement at all times. Keep learning. See you soon. Take care and bye till next time.